I grew up in British Columbia and spent a lot of time outside, and that's where I developed my curiosity for the natural world. So the opportunity to come back here to do research that may have implications for the long-term sustainability of the system was an incredibly enticing opportunity. It's kind of a dream come true for me. My name is Dylan Shea, and I'm a PhD student at the University of Toronto. On board the Martin Sheen this season, I've been collecting water samples at active salmon farms as well as at control sites throughout the Broughton Archipelago and Discovery Islands regions. The goal of this research is to determine whether salmon and fish in general experience an elevated risk of encountering viruses and bacteria when migrating past salmon farms. So we're collecting water samples and filtering out all of the particles so that we can subsequently genetically screen those samples for the presence of infectious agents. One of the groups of infectious agents that we're screening for are viruses. And in particular, one notable virus that we're screening for is piscine reovirus. Piscine reovirus has been associated with a condition called heart and skeletal muscle inflammation, or HSMI, in some species of fish, which is particularly important for migratory species as it affects the working muscles of the fish and can have extremely detrimental impacts on migratory success. I hope that this research contributes to the conservation of Pacific salmon so that generations to come can see the beauty that is British Columbia and all the species that rely on Pacific salmon for survival. Without the help of the crew of the Martin Sheen, I would not have been able to do surveys of the Broughton Archipelago and Discovery Islands. So I've been incredibly fortunate to have all the help I've had this season, and I'm very thankful that Sea Shepherd came here to investigate this issue.